And so I know as we get more folks who are joining the session, they won't be able to see the chat history. So I will periodically keep posting this link for the digital attendance form. So make sure you fill that out. You only need to fill it out once. So if you've already clicked on that link, and then you are good to go. But just as people keep kind of dropping into the session, I will kind of periodically post that into the chat. Okay, well, it's 4 p.m., so we might as well get started. I'm going to now share my screen, which is going to show you the PDF of this lab exam guide. And we're just going to go through it together and talk about these different lab exercises. You know, what were the most important points to kind of glean from each exercise, and what are the types of questions you can expect to see on this lab exam. So feel free to, you know, turn on your microphone if you have a question, if you want to stop me, or you can put it in the chat. I'll keep an eye on the chat. So if you type something there, I will see it. So, all right, we're actually starting not with our first lab exercise, but with lab exercise 3, which is about microscopes and kind of the basic principles of microscopy. So you will have a few questions on your exam where you'll need to identify parts of the microscope. So you want to be comfortable with labeling each of these parts and then identifying its function as well. So let's go through this so we know what each part is. So A, what is part A? You can either speak into your microphone or you can type it into the chat.
Ocular lenses.
Right, so those are the ocular lenses. Oh, thank you, Chanel. So that is the part that you're actually putting your face up against to see your sample. And then if we move down the ocular lens, we get to a different type of lens. What do we call these?
Objective lens.
Right, yeah, those are the objective lenses. And the objective lenses sit on part C, which is like a little turnstile piece.
Those pieces.
What was that?
Nose piece.
The nose piece, yeah, exactly. I see that we have a few more people who have joined us, so I'm going to post the link again to the sign-in sheet. If you've already signed in once, you don't need to do it again, but as people keep joining, I will keep posting that in there. Okay, D.
Mechanical stage.
Yeah, D is the stage where we can put our sample. Then we've got E, which is pointing to this mechanism here that's like underneath the stage.
That stage adjustment knob.
Yes, yeah, that is the stage adjustment knob. So that is how we can move the stage backwards, forwards, left, right. So we can kind of view all around our sample. All right, and then F and G are pointing to these knobs. It's a little bit grainy, these images, not great resolution. But F and G are these two large knobs, so which is which? They kind of do the same thing.
F is the fine adjustment and G is the coarse adjustment.
Right, yeah, so G is like the larger outer knob. So that's for the coarse adjustment to move the stage very rapidly up and down. And then F is the finer adjustment.
This is Heather speaking. How can I help you?
For the fine focus. All right. H and I, these are two parts that often get mixed up by students.
I'm not sure if they're the ones that would assist, but if not, they should know who they need to speak. Okay? All right, no problem.
So what is H?
Is illumination.
Yeah, so H is our light source. And you often see the condenser on there as well, right above the light source. Or actually, no, I'm sorry, the condenser is up. That's I. I is our condenser. Okay, and then finally J and K. So J is pointing to this kind of metallic part on top of the stage. Right, Kadiatu, that is our frame. It's also called like a stage clip. It's like a slide frame, but it basically keeps your slide in place.
Professor, I have a question.
Yeah.
Dr. Smith said that I was called the iris diaphragm wheel, which was not the condenser when he was explaining it to us. So I just wanted to clarify. Like he said that the condenser was separate from the iris diaphragm wheel.
Right, they are two separate things, the iris diaphragm and the condenser. And since it's pointing to like this lower part, that's what I identify as the condenser, and then like right above it is the diaphragm. I mean, it's really hard to tell in these images. So I mean, I certainly don't want you to get confused though if Dr. Smith said something separately.
So I mean, if like that was something we were tested on and it like was clear that it could be either one based on the picture, would you grade it either way?
I would like I guess it would depend on how the question is worded, but I do like give partial credit if I can see where a student kind of misunderstood the picture or like misinterpreted the picture. So I can't speak for all professors, obviously. But yeah, so it's so hard to see because this picture is really grainy, but the condenser, okay, so here in I, now I'm seeing it is like pointing towards a wheel. So I can see why he would say it's the iris diaphragm wheel, because that is like a moving part, whereas the condenser is like something that you don't adjust. It's kind of static. I actually, when I was doing this review, I identified H as the condenser and then I as the iris diaphragm. But then in the official like answer key, it said that H was the light source and I was the condenser. So that is a discrepancy that I think we need to rectify amongst the biology 150 teaching faculty. So I guess, yeah, I would identify this as the condenser. I guess some faculty are saying it's the iris diaphragm. Let's see, just out of curiosity, I want to bring up that the exercise 3 where you had this labeling, because I think the pictures are clearer on that. Since we do have a little bit of a lull, and I noticed some more people have showed up to this review session, please follow this link to sign in and record your attendance. Just a moment, I have to stop my sharing in order to open up this file. All right, so let me share this with you because this gives us a lot more detail than what we have in our review packet. So this is directly from that exercise 3 lab handout. So all of you should have a copy of this somewhere on your computers, or maybe you have it printed out. So here it is showing underneath the microscope. Okay, so this is our light source, which is kind of in the base. And then underneath the stage, you have these layers of rings, basically. So the bottom one is the condenser, this kind of smooth one. And then the wheel, the wheel with the ridges on it, that is the iris diaphragm adjustment. And so you can see it actually has like numbers to correspond to the different objective lenses. So if I'm pretty sure these are the kinds of detailed images that you'll see on your lab exam. So not like the super low res ones that are in the review packet, but the ones that are in the actual lab handout. So these images are obviously a lot more clear and have a lot more detail in them. So I would just really pay attention to the difference between these two parts. So the condenser is like this smooth part, and the iris diaphragm is this sort of ridged turning wheel thing.
Okay, thank you, Professor.
All right, so I'm going to bring back the review packet and we can move on. Do we need to go over any of the functions of these microscope parts? I mean, we're pretty clear on like what the lenses do, what the power button does, what the focus buttons do, or the focus knobs. All right, if we are good to go, then I will just keep on scrolling. Let's talk about question number 2. So if we are going from a low to a high magnification, so let's say from a 4x to a 10x, what do we expect to see happen to the size of the image, the intensity of light, and our field of view? So let's imagine we're going from 4x to 10x. Is the size of the image going to get larger or smaller?
The image will get bigger.
So the image is going to get bigger, yes. The size of the image is going to be bigger because if you are going into from 4x to 10x, you're zooming in. So you are going to make that image larger than it was previously. So the size of the image is going to be bigger. What's going to happen to the intensity of light? This one's a little harder to answer because you haven't had the chance to actually physically use a microscope in class. But what do you think would happen to the intensity of light?
It's less light.
That is correct. So the intensity of light is going to decrease. The field of view. So how much area can you see through your lens? Is that going to get bigger or smaller?
Smaller.
Right, it's going to get smaller. Yeah, so as you zoom in, you're going to see more detail, more of your sample, and you're kind of reducing that field of view. Okay, what about resolution? So that means the amount of detail. Are you going to see more detail or less detail?
More detail. Less detail.
Well, if you're zooming in, if you're going from lower to higher magnification, you are expected to see more detail. You know, think of like that picture of the flea in the lab exercise. At 4x, it's kind of hard, you know, you can kind of tell some of the legs and stuff like that are there. But when you really zoom in, then you can see like the individual hairs on the legs and the detail in the face. So the resolution is going to increase. As long as your yeah, as long as the focus is there, you might need to adjust the fine focus a little bit, but the resolution should increase. And depth, I'm not sure what it means by depth. I guess it means like literally like how far the objective lenses are going.
The other teacher said the depth of field decreases.
Depth of field. Okay, yeah, I can see that. So yeah, depth of field increase. And yeah, we did resolution. Okay. So what do you think are the advantages or disadvantages of observing a specimen at like low magnification versus at higher magnification?
You'll be able to find your specimen, the whole thing of it, you can see it.
Okay, yeah, so at low magnification, we can see the entire sample. Yeah, so I guess we can see, well, yeah, we can see like the big picture of things. And then what about for higher magnification? What's the benefit there?
We can see like the smaller parts of our sample.
Yeah, okay, so see the smaller details in the sample. And that's, I think, the best way to sum it up, that it depends on what your needs are, whether you want to see kind of the whole context of your sample, or you really want to hone in on exactly what's going on in one particular, I don't know, area or cell. So there are advantages and disadvantages of both. All right, this next one, this is important. You're definitely going to have questions like this on your lab exam, where you'll be given some values and you'll have to calculate the field of view at a different magnification. So remember, this is referring to that C1 V1 equals C2 V2 equation. So if you know how to use this equation and you know how to set it up given this information, then you're in good shape. So what would be our C1 V1? Oops, sorry, I'm using the wrong values. M1 D1. M2 D2. All right, so M refers to magnification, right? So how are we going to set up this equation?
$40 times 6,000.
Right, so 40x times our diameter at 40x is 6,000 microns. We'll do 40 times 6,000. And then we want to try to find the field of view at 400. So 400 times D2. So then we'll have, is it 24 divided by 400 equals D2. And that comes out to 600 microns. Another way you could do this, if this equation is a little more intuitive to you, that if you're going from 40 to 400, if you're increasing your magnification 10 times, that means you're decreasing the field of view also by a magnitude of 10. So all you need to do really is cross out that last zero and you have your answer. So that is the kind of quick way to do it. I know oftentimes it'll ask you on the lab exam to show your work and show the calculation that you use. So it is good to know how to do it kind of the long way. So then you can show your answer and not just type out 600. You want to show your work, show how you calculated it. All right, how about these conversions? Do we need to go over these conversions at all? How are we feeling about those?
Can we actually review them, please?
Yeah, absolutely.
Yeah.
So typically, for the most part in the metric system, we're talking about when we go from one unit to another, it's an order of 1,000. So if you just memorize kind of what's bigger and what's smaller, and you know that 1,000 rule, converting becomes pretty straightforward. So here we have 3 millimeters, and we want to go into micrometers. So is a micrometer smaller or bigger than a millimeter?
Smaller.
Yeah. Exactly, yeah, so it's smaller. So if we're going into a smaller unit, we know we're going to have more of a larger number, basically. So we have 3 millimeters, and just trying to move this over here. So we have 3 millimeters, and then we are going to multiply that by 1,000 because there is 1,000 micrometers in a single millimeter. So 3 times 1,000 becomes 3,000 micrometers. So we're going from a larger unit to a smaller one. Larger unit means a smaller number. Smaller unit, you have a larger number. So now let's go in the opposite direction from micrometers into millimeters. So we know that this is smaller. So we're going from smaller to larger, the units. So now instead of multiplying by 1,000, we're going to divide by 1,000. So what's 500 divided by 1,000?
0.05.
You added in a 0 where it shouldn't be, or maybe you just misspoke, but it would be 0.5, 0.5, because basically we are just moving the decimal place over 3 places. And so that is 0.5. So half a millimeter is 500 micrometers. Millimeter, micrometer. Okay, now this is also along the same vein. So millimeters, 0.25 millimeters is how many micrometers?
250.
Exactly, yeah, 250. All right, and now we're going to move from micrometers into nanometers. So a nanometer is even smaller than a micrometer. So how many micrometers is 4,000 nanometers? Oh, Ryan's got the answer. It would be 4. 4 micrometers equals 4,000 nanometers. All right, very good. There is some conversion, I believe, of units, of metric units. So it's always a good refresher to remember kind of what the order of the units are, you know, what's bigger, what's larger. Once again, I'm going to post this link in the chat in case anyone has arrived a little bit late. So if you haven't already signed in for the digital attendance, make sure you do that. All right, now we're given a little image of these Elodea, these aquatic plant cells. So it tells us it's at 1,000 magnification. And this is using a microscope that has a field of view of 5,000 micrometers at 40x magnification. And so we are to use this information to determine the size of these cells. So given these numbers that they're provided for us here in the question, it's clear that the first thing we need to do is figure out the field of view. You know, how far is this across? So we know what the field of view is at 40x. So now we have to calculate what it is for this image at 1000x. So again, we're going to use that M1 D1 equals M2 D2 formula. So we'll have our magnification 40 times the field of view at 40x, 5000 equals, the other magnification is 1000, and then we are solving for this D2.
The 200?
It is 200, yes. Yeah, so the field of view here, what we're seeing, this image is 200 micrometers across. So if this entire circle is 200 micrometers across, roughly how long is each of these cells?
Well, if you assume there's 2 cells per length for field of view, wouldn't it just be 200 divided by 2 equaling 100?
Yeah, that's a very quick way to kind of, estimate it. So yeah, if we're looking at it across, so if we have our, if we know that this diameter straight across is 200, and yeah, we have about 2 cell lengths, give or take, yeah, that would be a perfectly reasonable estimation. So our diameter field of view divided by how many cells across. So they are about 100 micrometers long. And then what about the other dimension, this width? So that's a little bit trickier. Let's see, how many widths do we have? 1, 2, 3, 4, maybe 5. So 200 micrometers divided by about 5 widths. That's what, 40 micrometers wide. Wonder if I can turn this around. Nope.
I'm sorry, Professor. Can you repeat why you divided it by 5 again?
Sure.
Oh,
So
it's
this was asking us for the length and the width of the cells.
Okay.
So the first dimension, the length, that would be like the longer part of the cell. And so that one was pretty easy because it's roughly, 2 across. But then for the narrower dimension, I was just kind of counting how many widths I saw, like going from one end to another. So I saw 1, and then 2, 3, 4, 5. So yeah, we have about like 5 cells wide on this field of view.
Okay. Yeah. Okay. I got it. Thank you.
Mm-hmm. So what we just used, this was the counting method of kind of estimating lengths. What was that other method for estimating how the size of images or size of things under the microscope?
Like percentage?
Mm-hmm. Exactly. Yeah, there's also the percentage method, which, in certain circumstances, that's more convenient to use than the counting method. I guess in this case for width, we could say, oh, it's about, this one cell takes up about 50% of the total diameter, right? So that would be 200 times 0.5, which again would give us the same estimation, 100 micrometers long. And then if you're saying, well, you know, this width is about 20% of this total view I see. So that would be 200 times, is that right, 0.2? Yeah, and that would also give us 40 micrometers wide. So, you know, two different ways to come up with about the same answer. All right, so that does it for the microscopy exercise. Are there any other questions about microscopy before we move on to the next part of the review?
Yes, Professor, I have one question. So could you just quickly explain how is it that you knew to use the M1 D1 equals M2 D2 for number 6?
Sure. So when you're talking about calculating the field of view, typically it's really hard to measure the field of view at a very high magnification. So it's easier to measure it at a lower magnification and then use this kind of ratio formula to calculate what the field of view would be as you increase your magnifying power. So this is just the kind of known way to, if you have the field of view, if you know what your diameter is at a low magnification, you can sort of convert it to a higher magnification using this formula.
Okay, thank you.
Mm-hmm. So yeah, typically if you see a question and it gives you this information, like it gives you magnification, two different magnifications and only one diameter of a field of view, that's kind of a hint that you're meant to find the other diameter of the field of view. So it's you know, it's giving you 3 out of these 4 values. So that's an indicator that you're supposed to find that 4th value. And now this question actually had the extra step where it wasn't just calculating the diameter, but then you had to use that information to estimate the size of this of these cells. So that's another thing is make sure you read every question on the exam really carefully and that you understand what it's asking you. Because if you only read the first part of this question, you might assume, okay, I came up with 200 micrometers, I'm done answering this question. So it's easy to miss this last part where it tells you that, no, this is just the first step. Now you have to use that to find the dimensions of these cells. So don't overlook any part of these questions. Even if you think you know what is being asked, just you want to, you know, double check and make sure that your answer is corresponding to what was asked of you. Okay,
Professor, for us to show our work, is it going to have like a typing dialog box for us to write all of that in it as the answer?
Yes, yeah. So for these types of questions where we want to see some values and calculations, it'll be, yeah, like a larger kind of text box for you to type into. Because this exam, it's going to be over Blackboard again. So now you're kind of familiar now with the Blackboard kind of exam format. So for a question like this, you would have like a large kind of text box and you would have like the same like format toolbar at the top if you wanted to, you know, indent or change.
Kind of like the free response text box that we usually get.
Exactly. Yeah.
Okay.
Exactly. All right. So let's talk about, this is all the way back, our second lab exercise about organic molecules. So what are the 4 classes of organic molecules?
Proteins.
Carbohydrates, protein, lipid, and nucleic acid.
Yes, protein, lipids, carbohydrates. I'm sorry, it was hard to hear you at first. Carbohydrates and nucleic acids, yes. Everything on Earth is made up of just one or a combination of these four things. So these are our major classes of organic molecules. A monosaccharide, what is it? How would we define that?
It's the simplest form of sugar.
Right, so it's the simplest form of sugar. Another way you could word that is it's the monomer of a carbohydrate. So putting together many of these monomers, you could come up with a polymer like a starch or something. Ooh, what are reducing sugars? Who remembers what that's all about?
Are the reducing sugars the disaccharides?
So all monosaccharides and then some disaccharides are reducing sugars. Well, what does that mean? That refers to something in the structure of these molecules.
Carbonyl.
Carbonyl, yeah. So reducing sugars contain a functional group called a carbonyl. Who remembers the kind of chemical makeup of a carbonyl group?
Is it carbon double bonded to oxygen?
It is. Very good, Karen. Yeah, it's C double bond O. That is our carbonyl. So reducing sugar is, a sugar molecule that has a carbonyl group that is able to donate electrons. And that brings us to the next question. So reduction, what is reduction?
You gain an electron.
Yeah, reduction is gain of electron. And so by extension, oxidation is the loss of an electron. So a reducing agent, this is something that allows the reduction of something else. So the reducing agent is donating the electron. And along the same theme, oxidizing agent. Oh, thank you for whoever reposted that link for Jenna. The oxidizing agent retrieves or accepts the electron. Okay, and one way to remember the difference between oxidizing and reduction is oil rig. So oil rig, oxidation is loss, reduction is gain. So if you can remember oil rig, then you'll never be confused as to what reduction and oxidation mean.
Professor, I have a question.
Mm-hmm.
So we are talking about reducing and the reducing sugar. The reducing sugar, we call it reducing, is it because the carbonyl group can gain an electron or donate?
It is a reducing sugar. It's because it's a reducing agent. So it is donating the electron. So it itself is being oxidized, but it is a reducing agent. Yes, this is where language can be very difficult because it's easy to get confused. So reducing sugars, like any reducing agent, a reducing agent is what is giving up the electron to whatever is being reduced. And likewise, the oxidizing agent, that is what is accepting the electron.
Okay, so the reducing sugar gain electron, then they become oxidized, but other sugar which has no carbonyl group, they cannot be oxidized, right?
Wait, reducing sugar. wait, you said the reducing sugar is, I don't know, hold on, I think you, I'm getting mixed up now too. The reducing sugar, the carbonyl, this is what's going to be, this is donating an electron to something else. So the reducing sugar is actually going to be oxidized.
So when something is being oxidized, then it means that they themselves are the reducing agents. And then when something is being reduced, then it means that that molecule itself is the oxidizing agent.
Yes, yes,
Okay.
Diana, you got that perfectly correct. And I think you explained it well too. I'm going to try to write that out here. So yes, the reducing agent is oxidized. So it's losing the electron because it's donating it to another molecule. And then an oxidizing agent is reduced because it is accepting an electron and it's gaining one. So I apologize to non-native English speakers. I'm sure that this is confusing for you. It's just as confusing though if you grew up speaking English. It's not intuitive. It doesn't make sense just if you don't know exactly what these terms mean. So it's important to remember, yeah, what oxidation is, what oxidizing means, and the difference between those two words. It's a nuance, so it's difficult, but just try to commit this to memory so you don't get confused.
It's funny that you say that, Professor, because I feel like whenever we review oxidizing and reducing and what is oxidized and what is reduced in class, I always feel like we're being told like white is blue, but blue is actually white.
Yeah, yeah,
Yes.
like reducing, that should indicate that something is you're losing something. But yeah, the fact that reduction is a gain of electron, yeah, I've always felt that was a cruel trick played on all of us by chemists.
Yes, for sure.
Professor, I get this sentence. It does not confuse me at all because we call it reducing. Someone is reduced because it gain electron. When they gain electrons, so the charge is reduced, like from 0 to -1, -2. So understand that part reduced. Why it is reduced? Because the charge is reduced, and so why I'm asking that question is I'm trying to connect the carbonyl group to the reducing sugar. So reducing sugar and something is reduced. So I'm wondering if it's because the carbonyl group will gain electron in the reaction. That's why we call it reducing sugar. Am I clear?
I see what you're saying. Okay, so you are just confused about what's happening with the carbonyl group and how it's
Yes.
reducing. Okay. Let's see. I have a visual for that. Let me see if I can bring it up.
But it's fine. I don't want to waste other classmates' time, so it's not really related to this review.
Okay. Well, I mean, I
But I can-
appreciate that you are curious about it. And, you know, also if it helps someone else understand it better, then I think it's worth it to bring it up. When I'm trying to find...
I don't want to confuse everyone, but thank you for the explanation.
Guess I don't have an image of it. Okay. All right. So we'll move on then. Okay, so speaking of these reducing sugars, we used these two assays, the Benedicts and the Barfoids assays, to identify what was a reducing sugar. So these two reagents, Benedicts and Barfoids, they're really similar. They even look the same, right? They're like this light blue color, but then they reacted with different molecules. So why did they have different capacity to react with things?
Do they have a different pH?
So
They do have a different pH. Yeah, can we guess which one is, I don't know, which one is more acidic than the other?
the Barfode?
Yes. Yeah, the Barfoids is has a pH of about 4.5. And so the Barfoids was only able to interact with monosaccharides and not just all reducing sugars. So remember, Benedicts can react with monosaccharides or reducing disaccharides, but the Barfoids can only interact with the monosaccharides. So if we perform this Benedict's assay with glucose, we know glucose is a carbohydrate monomer. So what is being oxidized and what is being reduced? So oxidized, what is losing the electron, and reduced, what is gaining the electron? All right, glucose. Very good. Yeah, glucose is losing its electron. And then what's being reduced? What was it in these solutions that was actually reacting with the carbonyl group?
Oxygen?
It wasn't oxygen. So there was an oxygen component with creating that kind of red precipitate in the tubes.
Copper hydroxide.
Yeah, it had to do with cupric hydroxide. So it was CuT plus 2+, which got reduced to Cu3+. So going from a cupric to a cuprous ion. So yes, so this is what was chemically happening in that Benedict's assay. And speaking of this assay, so we had all of these samples in solution. So like glucose, so we just had some sugar that was dissolved in water, because water is our universal solvent. And in a solution, glucose exists in both a chain form and a ring form. So what does that exactly mean? How can we explain that?
That the chain forms have the ability, I guess, to bond with other molecules, and the ring forms don't because they're closed.
That's actually getting towards the answer for number 14 of why some molecules are reducing sugars and some are not. But before we get to that, let's think about this glucose molecule. So we know it's usually depicted as like the 6-sided ring, or it can be in sort of a linear form, right? And so it's going to switch back and forth between this ring form and this linear form. And what is directing it to switch back and forth between these two shapes? So the answer really is that nothing tells it to take the ring form over the linear form. If it's in solution, if it's dissolved in water, it just kind of spontaneously switches from one form to another. And so you'll have roughly these two forms are in equilibrium. So at any given time within your glucose solution, half of the molecules are going to be in the ring form and the other half are going to be in a linear form. So it's just kind of a random, you know, how these molecules like to arrange themselves, but it's a spontaneous process going from one shape to another.
Does it have to do with losing the electron from the carbonyl group?
Yes, it is related to that. And that's actually getting to what Diana was saying, which I said was similar or kind of getting closer to this question or the answer for question 14 of why some sugars can be reducing agents and others are not. So this question of why is sucrose not a reducing disaccharide has to do with the availability of that carbonyl group. So yeah, fructose and glucose, when they're on their own, individually, they're reducing sugars, but when they come together to make sucrose, suddenly they don't have that reducing capability anymore. That carbonyl group sort of still exists, but it's not going to be able to do any reducing. Oh, I'm sorry, no, the carbonyl group is going to be used to form the bond between those two. So sucrose, we did have that in our assays. That was not one of the reducing disaccharides, and it's because that carbonyl group, it basically disappears when you're bringing together a fructose and a glucose molecule. So that carbonyl group kind of becomes the basis of that glycosidic bond, that covalent bond that links the two monomers together. All right, let's move on to, it's asking you for all the monosaccharides and reducing disaccharides that we assayed in the lab. So this actually requires you, it might require you to go back to that lab exercise and look at that chart that you filled out. So I know glucose was one of them. What were some other ones? I believe there were 3 monosaccharides and 2 reducing disaccharides. Okay, so benzoate, oh, I see Reem, you've also put in the answer. So yeah, we've got glucose, fructose, and galactose. So those are all monosaccharides. And then our reducing disaccharides are the maltose and lactose. Exactly. So maltose and lactose, those are 2 disaccharides that can donate an electron. Sucrose is a disaccharide that cannot. So we're talking about there are 2 different like functional abilities of these disaccharides. What is the difference structurally between these 2 kinds of disaccharides? So we just mentioned it. What is the structural difference between something that can reduce and something that cannot?
Carbonyl group.
Right, yeah. So the non-reducing disaccharides do not have that available carbonyl group. So it is a kind of, well, a functional difference or a behavioral difference of the molecule that is defined by a structural difference. So moving on from disaccharides to larger polymers, the polysaccharides. What are 3 polysaccharides we learned about, and what are they used for in cells? Starch, very good, Kariatu. Cellulose, yep, another good one. Oops, cellulose. So what are the functions of starch and cellulose?
Storing glucose.
Yeah, so starch is a storage form. It's a storage polysaccharide. So it stores glucose. What about cellulose?
Structure.
So we know it's in plants. Oh, I think I heard someone say it's structure. Yeah, so this is a structural polysaccharide. So plants have starch. What do human cells have, or animal cells? It's our storage of glucose.
Glycogen.
Right, exactly, glycogen. Yeah, so starch and glycogen, those are our storage polysaccharides. Storage of glucose, which we know is, our fuel, our source of energy. And then cellulose is the structural polysaccharide. So it's for making that rigid cell wall. If you took a starch molecule, and you completely hydrolyzed it, means if you performed hydrolysis on all of the covalent bonds holding those glucose molecules together, yeah, you would just get a bunch of glucose. So hydrolyze, that is the verb form of hydrolysis. So remember, hydrolysis is when you break a bond with water. So hydrolyze means you are breaking the bonds in that starch molecule with water. And you would end up with just a bunch of glucose monomers. So these next few questions could get a little bit tricky. We really got to remember, got to keep in mind what these different assays indicated and how the results, at least of the carbohydrate assays, are related. So if we had a substance, because there will be questions like this where you're actually shown the results of these assays, and you'll be asked, like, what kind of molecule is this, substance X? So if it gives a negative Benedicts and negative Barfoids, positive iodine and negative Biaret. So the only thing positive here was the iodine assay. So what does that indicate?
Starch.
Right, yeah, so that would be our starch. So now we have positive Benedicts, positive Barfoids, negative iodine, negative Biaret. So we know it's a carbohydrate of some kind because we got positive Benedicts and positive Barfoids.
Glucose.
So so it would be glucose. It would be the positive Benedicts would tell us that it is what?
A monosaccharide?
That's what the Barfoids would tell us.
Oh,
What about the Benedict's assay?
That it's a reducing sugar?
Yes, the Benedict's assay means it's a reducing sugar. And then if it's a reducing sugar and we also get a positive Barfoids, that's what tells us it's a monosaccharide. So yes, that would be, and that lines up with what glucose is. It's a monosaccharide, and we know it's a reducing sugar. So Okay, if
So
we have
it
a
can be fructose, right?
I'm sorry?
So it can be fructose or galactose.
Yes, theoretically, yeah, if fructose or galactose was also in this drop-down menu, or if it just said monosaccharide, that would be also correct. So if we have positive Benedicts and then everything else is negative, well, we establish that positive Benedicts means it's a reducing sugar. So if we look at our options,
Would it be maltose?
Maltose.
right, yeah, maltose. So maltose is one of those reducing disaccharides. So it would give us a positive Benedicts and then the negative Barfoids. All right, substance is positive in the Biaret assay, negative in all others. What was the Biaret assay for?
Protein.
Right, for protein. So let's see which one of these is a protein.
The egg.
albumin, yep. And then if all assays were negative, what might that tell us that we have?
Sucrose.
Right, sucrose. So remember, sucrose, this is a non-reducing disaccharide. So, I mean, if we had an assay or if we had a substance and all the assay results were negative, it's either water or just like, the absence of something, or if it had to be one of these choices, the only one left that makes any sense is sucrose, because it would not test positive in any of these assays. All right, do we want to go over 24 and 25? I heard a yes. That's good enough for me. Okay, so Benedict's assay. Jenna, what do you think? Benedict's assay.
Reducing sugar?
Yeah, so this is for reducing sugars. They could be monosaccharides, they could be disaccharides, we don't know. Positive reaction produces what kind of color?
Red.
Yeah, it's sort of like a red, brown precipitate. Our barfoids, so that's the other carbohydrate assay. What does it distinguish between?
Reducing sugars?
Yeah, so the Benedicts is for reducing sugars. And then if we had a positive in Benedicts and we put it through barfoids, what does that let us know? What kind of
Monosaccharides or reducing disaccharide.
exactly? Yeah, it tells us whether it's a monosaccharide or a reducing disaccharide. Because only the monosaccharides would be positive in the barfoids assay. All right, what was our protein assay called? Also started with a B.
Biaret. B-I-O-N.
Yeah, Biaret. I've also heard people call it Biaret. All right, if it's positive, what color does it change?
Is that the one that turns like purple?
Yeah, it turns like a nice lilac-y color, purple. And what does this positive result indicate? Or what's actually being detected in the proteins?
Peptide bond.
Exactly, yeah, the peptide bonds in between those amino acids. All right, so you will have some questions on your lab exam kind of related to the scientific method, where it'll tell you or it'll explain sort of a fictitious experiment and then ask you, to develop a hypothesis or to identify variables. So you do want to be pretty comfortable with this kind of activity. I think the most difficult thing for students is writing a good hypothesis. The other stuff I think is pretty self-explanatory, like, you know, the independent and dependent variables, or the experimental and control groups. But being able to identify or to write out a hypothesis, I think, is what people struggle with most. So let's try to do that here with this. So, okay, we have this fictitious company Y, that wants to test a new chemical to act as a sunscreen. So they're going to test it. They're going to apply this lotion to various volunteers in a certain area on their arms, and then put them out in the sun and measure amount of redness every 5 minutes for a total of 30 minutes. And then each person has 3 different lotions. So a lotion with their new chemical, a lotion with the currently marketed chemical, and then a lotion that has no active ingredient at all. So let's start with the question being asked. So a question is something really broad, really general. Hypothesis is more specific and like honed in on the details. So in general, what is the question being asked?
Which sun lotion will prevent sunburn?
Yeah, that's good. Which lotion or like chemical prevents sunburn? Sunburn better or more effectively. Now, what about a hypothesis? So a hypothesis has to be specific, has to be testable and falsifiable. So how would we word a hypothesis that's being tested here?
The new sunscreen chemical Sun X prevents sunburn better than sun-kissed lotion.
Okay, we're on the right track. So prevents sunburn better than sun-kissed. So let's take a look at this statement. Sun X prevents sunburn better than sun-kissed. So this word better, it's kind of ambiguous, right? It's not specific enough, because better is kind of a relative term, and we want to deal with something that is testable, something that's measurable. So how do we measure if it's going to be better?
Sun X reduces redness of the skin.
Excellent. Yeah, reduces redness of the skin. More than sun-kissed. Yeah, so we're getting closer. So prevents redness of the skin. Now we want to add as much detail in here as we can from this little description. So we know that they are measuring redness of the skin. What kinds of conditions are they, under which are they measuring this? So they're looking at redness of the skin after applying these lotions, and then what else do they do?
Over 30 minutes.
Okay, yeah, over 30 minutes.
When in direct sunlight?
Yeah, when in direct sunlight for 30 minutes. Now that is specific, it's testable, it's falsifiable, and it kind of brings into play all of these details that were mentioned here. So Sun X prevents redness of the skin more than sun-kissed. I guess when subject is in direct sunlight for 30 minutes. Yeah, that's good. That's a really good hypothesis. That's solid, because it's bringing in the relationship between variables, and it's something that you could test. You can design an experiment based on that hypothesis and say, yes, this hypothesis is supported, or no, it's not supported by our evidence. All right, so it wants you to make sure it's testable and falsifiable. Yes, it is. So what about our variables for this experiment? What's the independent variable? What's the dependent variable?
The independent variable would be the sunlight exposure for 30 minutes.
So yeah, I guess one of the independent variables, one of the things that we are controlling here is time in the sunlight. Another one is what? Yeah, type of sunscreen. Thank you, Reem. Type of sunscreen chemical. And then the dependent variable is what we are measuring.
Which would be the redness of the skin.
Skin redness, yes. All right, Jenna asks if we could explain the difference between these variables. So our independent variable is what we manipulate as researchers, as scientists. So we can determine how much time these subjects are spending in the sunlight. And we're also going to determine, you know, which type of chemical they have on their skin, or I guess we're putting all three on the person's arm, maybe like 1, 2, 3, like all in a row. So you can really compare the redness. So that's, again, if we wanted to test like 5 different chemicals, we could do that. If we just wanted to test 2 chemicals, we could do that. So we get to determine which chemicals we're testing, how many of them we're testing. So that's why it's an independent variable. This is what we are manipulating, and for the purpose of conducting this experiment.
Professor?
Yeah.
Just a quick tip for whoever asked that question. I struggled a lot identifying independent and dependent variables. And our professor told us a tip to, I guess, kind of make sense of it. So you could put it all in a sentence. So you would say the skin redness, or the redness of the skin, depends on the type, the time and sunlight, and the type of sunscreen. And for example, if you were to say it vice versa, like the time and sunlight depends on the redness of the skin, then it kind of doesn't make sense anymore.
Right.
So when you put it into a sentence and you say the skin redness depends on the type of sunscreen, then that way you know that the skin redness is your dependent variable and the type of sunscreen is your independent variable.
Okay, great. Thank you for that tip. And Jenna, thanks you too. Awesome. Thank you for sharing that, Diana. And yeah, I agree that that can be a quick way to kind of double check your answer and make sure it makes sense. That what you're measuring is dependent upon what you have manipulated or changed in your experiment. All right, so we had our 3 groups of patients. Which ones were the experimental group? I guess I'll scroll back up. So groups 1, 2, and 3, which were experimental, which was our negative control, and which was our positive control?
So group 1 would be the experimental group.
Mm-hmm.
Group 2, I believe, would be the negative control. And group 3 would be the positive control.
You said group 2 is negative with the sun-kissed, and group 3 is positive?
Actually, I think it's reversed. So group 3 would be the negative and group 2 would be the positive.
Right. Right, so our negative control would be the lotion that has no active ingredient in it, because that would show us, you know, what it looks like without any sunscreen protection. And then group 2 is a positive control because it's already known, it's already established that it works. So this would show us the effect of a known sunscreen chemical. And then the group 1, the Sun X, that's our experimental because we actually don't know what the results are going to be. So that's why it's good to have negative and positive control so that you know what an absolute disaster of a sunscreen result would look like. So if Sun X didn't work at all, then the skin would burn just as much as no sunscreen, right? Or if it works really well, it might work as well as our positive control, which has already been established that it is effective in this purpose. So I believe we have a graph here. Yes. So based on the graph, Sun X is more effective. Oh, is Sun X more effective than Sun-Kissed? And how does the graph show us, what the results are? How can we use this graph to come to a conclusion? That's what 36 is asking.
I think it would be, yes, it is more effective because the amount of redness goes from 0 to 25. So you can see that after 30 minutes in the sun, Sunkissed says that the amount of redness on the skin is at a level 5 compared to Sun X, which is still probably at like a 2. So it means that the skin is less red with Sun X.
I guess let's say quantifiably less. I guess it cut off the last part of it. Okay. But yeah, you're absolutely right, Diana. So here we have on our y-axis, it doesn't give us any units. I'm not exactly sure how they determined this, amount of redness, but regardless of what those units are, they have some kind of scale for measuring this. And clearly the least amount of redness was in the patch of skin that was treated with the Sun X chemical. So that would indicate that it is more effective than Sun-Kissed. So we got a little bit of redness with Sun-Kissed, even less with Sun X, and without any kind of active chemical or active sunscreen ingredient, there was a lot of redness after 30 minutes. And what would be a good descriptive title for this graph?
The effectiveness of sunscreen.
Okay, so effectiveness of sunscreen, that's a good start.
When exposed to direct sunlight for 30 minutes?
Okay, yeah, effectiveness of sunscreen when exposed to, oh yeah, I think direct sunlight for 30 minutes, yeah. Yeah, so your graph should, or sorry, your title should always tell the reader exactly what they're going to see in your graph. So a good way to start off is if you want to mention how these two variables are related to each other, so it could be effectiveness of sunscreen demonstrated by amount of redness over time or something like that. Or yeah, the effectiveness of different chemicals when exposed to direct sunlight for 30 minutes. But the more detail you put into a title, the better you'll be. So even if it sounds kind of like silly or long-winded, I would err on the side of putting too much detail into your title. At least that's what I look for as an instructor when I'm grading graphs. I like to see really, really good descriptive titles. So I would recommend that. All right, so we've been doing this for a little over an hour. How about we before we dive into osmosis, how about we take a 10-minute break? So it's 5:19, so let's start back up at 5:30. I'm gonna pause the recording and turn off my own camera so I can go grab a little snack, and I will see you all back here at 5:30. All right, it is 5:30 by my computer's clock, so let's get back to this review. Now we're looking at osmosis and diffusion. This was in exercise 4. And so these first few questions ask us about the relative tonicity. So we're looking at which beaker contains a solution that's hypertonic compared to the bag. So a hypertonic beaker solution. All right, we have a little drop-down menu here.
Is it 1, 2, and 5?
Yes, yeah, that is correct. 1, 2, and 5 are all of those beaker solutions are hypertonic compared to the solution inside of this bag. Yeah, so 0.6 is greater than 0.2, 1 is greater than 0.2, and 0.4 is greater than 0.2. So that is correct. Those are the hypertonic solutions. Now this question is worded a little bit differently. Which bag would you predict to show the least change in mass at the end of an experiment? So where would we
3.
expect to not see any net change? I see Rosa has provided us the answer. Yes, it is beaker 3, the bag in beaker 3 rather. So because these solutions are isotonic, there should be no net movement of water. Of course, there will still be water moving across that membrane, but the molecules going in are going to be equal to the molecules moving out. So there should not be any major change in the mass of that bag.
Professor, I was wondering if you could review just the definition of hypertonic.
Sure, hypertonic means there's more stuff dissolved in the liquid. So here we have the stuff dissolved in the liquid is usually numerically represented as molarity. So molarity is just how many molecules do you have per unit of liquid. So 0.6 molar versus 0.2 molar. And so if we're looking for what's more hypertonic, the beaker solution has a higher molarity, so it's hypertonic. It has just more stuff dissolved in it versus the solution inside here. So that's basically what it comes down to, is just comparing these molarity values. And so in this beaker number 2, once again, there's a higher molarity in the solution of the beaker versus the solution in the bag. So that's what makes the beaker hypertonic to the bag. And if we kind of switch the way we talk about things, we can refer to the same example, example 2, and say the bag or the solution inside the bag is hypotonic to the solution of the beaker because it has a lower molarity. There's just less stuff dissolved in the water inside the bag than dissolved in the water in the beaker.
All right, thank you, Professor.
Mm-hmm. So now we are arranging the beakers in order of the mass of the bags. So which bag is going to lose the most mass? Or basically, which is going to weigh the lightest? Which bag is going to weigh the lightest? Which is going to lose most water? So again, we're going to compare these two molarities to determine in which direction water is going to move. And then come up with an arrangement. Oh, okay. So how about, I kind of want to make some little like arrows or something. All right, so in this first example, is water entering the bag or leaving the bag?
Leaving the bag.
Water is going to leave the bag. Well, that could be a little bit bigger. Let's see. So if okay, well, that's my little arrow. And so this is indicating that water is going to leave the bag and enter the beaker. And that's because the water is being drawn out. The water wants to basically find an equilibrium where these two values will match. The ideal situation is where the concentration inside and outside on either side of this membrane is the same. So if it's trying to get to this kind of isotonic situation, then in order to dilute this 0.6 molar solution, we need to have water leave the bag. So water is going to exit the bag and enter the beaker. So this water is going to decrease in mass because it's losing water. Or I'm sorry, this cell is going to decrease in mass because it's losing water. And now beaker 2, it's a similar situation, right? 1 molar is greater than 0.2 molar. So any water that's in this bag wants to leave in order to dilute this more concentrated solution.
Oh.
So we are going to, again, have an arrow that's pointing out. So water is leaving the bag, or yeah, the cell or the bag. And then this is our kind of equilibrium situation. This is isotonic because the molarity on either side of the membrane is the same. So let's move on to example 4. Now we have a situation. Yeah.
Sorry, why do I think the water is going to go into the cell because the solution is hypertonic?
For number 4?
For number 2.
For number 2. Right, so the solution, the beaker solution is hypertonic compared to the cell solution, right? 1 molar, that's a greater value. That means that represents there's more molecules dissolved in this liquid than here. So if there's more molecules on this side of the membrane, the water wants to dilute those molecules. It wants these values to be about the same.
So it's not going to go into the bag?
No, water is going to leave the bag. Water is going to leave the bag for the beaker.
Okay, thank you.
Because if you think about it again, as in terms of like molecules that are dissolved in water, if water is leaving the bag, that means the remaining solution is going to get a little more concentrated. And if we have more water here, that means this solution is going to get a little more diluted. And that's what water is trying to achieve. It's trying to get to this kind of equilibrium where you have a more similar concentration of solutes on either side of a membrane. So now this example 4, this is the exact opposite of what we see in example 2. So now the solution inside the bag is 1 molar. The solution in the beaker is 0.2 molar.
Opposite.
Yeah, it's going to be opposite, exactly. So now water is going to enter the bag. It wants to dilute this very concentrated solution here. So let's just switch our arrow around. Oops, and put it that way. So yeah, now water is entering the bag. So this bag is going to gain weight. It's going to get heavier by the time we end our experiment. And then finally, number 5, well, we already established that this solution, this beaker solution, is hypertonic compared to the bag solution. 0.4 is greater than 0.2. So once again, we're going to have water leaving the cell or the bag and entering the beaker. So now that we know which direction water is moving, we can determine, which bags are going to be lighter, which ones are going to be heavier after 30 minutes. So it wants us to start with the bag that's going to lose the most mass. So there are 3 bags that are going to lose mass, 1, 2, and 5. These are the bags that are losing water to that surrounding beaker solution. So from 1, 2, and 5, which one do you think is going to lose the most mass, the most water?
2.
Yes,
2.
exactly. 2 is going to be losing the most. All right, so 2 is going to lose the most, and then which is going to lose the next most?
1.
1.
Exactly, 1. Oops. And then that leaves us with 5. So that's all the ones that water is exiting. Then next, we would say number 3. That one's, because that one's not really changing any mass. It has no net water movement. And then finally, number 4 is the last one, because this is actually going to gain mass. So the order of these bags from lightest to heaviest at the conclusion of an experiment would be 2, 1, 5, 3, 4. And we see that that's one of our answer options.
So, Professor, why did you list 4 there? Because the question is asking to list the bag that loses the most mass, right?
Right. So losing
So
the
why do
most
we have
mass
to
is
add
who is
4
going to
there?
weigh the least.
Oh, okay. I see.
And so number 2, this is going to weigh the least. It's going to lose the most water to the surrounding solution because the difference between these two values is so great. It's much greater than the difference in the other examples.
Okay, thank you.
Mm-hmm. All right, so when we move on to the next page, we see an explanation of a similar kind of setup where we have a bag filled with a sucrose solution that's placed in a beaker containing also some kind of sucrose solution. It tells us the bag's initial weight is 15 grams. After 15 minutes, it weighs 17 grams. And then it wants us to calculate the percent change in the weight. And then after 45 minutes, again, what is the percent change in weight? So how do we calculate percent change? What is that formula for calculating percent change?
Actual over initial times 100.
Almost, you almost have it right. So yeah, so the mass at some time minus the initial mass, and then divide that by initial mass. So you forgot about that subtraction there, but otherwise you had it correct. Initial mass. And then, yeah, we're going to multiply this by 100 to make it a percent. So, okay, so we have an initial weight of 15 grams. So our initial mass is 15. After 15 minutes, it weighs 17 grams. So that's how we're going to set up our first calculation. So the mass at time, that's 17 grams minus 15 grams divided by 15 grams. Oops. Times 100.
33.3.
23.3, that sounds about right. I have not done the calculation myself, so I'm going to trust you on that. So 13.33%.
It's 33.
Oh, 33?
Mm-hmm.
Do we agree with 33?
No, I think 33.3 is for 20 grams minus 15 grams.
Oh, okay. That makes,
And then it's 13.3%.
13.3.
all right. Yeah, so 13.3 is for the 17, and then that other one was our cumulative percent change after 45 minutes. Got a little bit ahead of ourselves there, but I'm going to set it up the same way. So our weight at whatever time point, which is 20, minus our initial weight divided by initial weight. And then don't forget to multiply it by 100 so that we get our percentage. All right, very good. This is one of the formulas that you'll have to know for the exam. It's not given to you, so you want to memorize how to calculate this percent change. And then, of course, not just memorizing it, but also knowing how to apply it, when to use it. But you'll see, questions similar to this one where it explains to you an experiment and it gives you certain values, and then it's up to you to know what to do with those numbers and how to calculate your answer. All right, now we have some solution dilutions. Starting with a 20 molar stock solution, and then we have to make some dilutions of different concentrations. So a 0, 2.5, 5, 7.5, and 10. And we know we want to make 50 mils of each of these dilutions. So this is where you want to use that C1V1 equals C2V2 formula. So concentration and volume of a known substance or what you want to make, and then C2V2, that's concentration and volume of a corresponding solution.
Professor, wouldn't it be M1 V1 equals M2 V2, or is that a different formula?
The M and D, like that's the one that we used. So this is basically identical to the M1 D1 equals M2 D2 that we used for calculating the field of view, because it works on the same principle that these amounts are related to each other. So just like when we're going from a low magnification to a higher magnification, and the diameter of our field of view changes in proportion to that change in magnification, very similar here. It's like we're magnifying or diluting a solution. So we're going from a very concentrated solution to a more diluted solution. So the amount of that initial solution is going to, the amount we need to make these different mixtures is going to change in relation to, what we're making. But I mean, there's a reason why these formulas are identical, because the changes are proportional, basically. But yeah, the C1V1, C2V2, just different letters to represent concentration and volume rather than magnification and diameter. But it's basically about things changing in proportion. So our C1, our concentration, our starting concentration is this 20 molar. Oops. V1 is the volume, the amount of 20 molar that we want. This is our unknown. This is what we're solving for. C2, this is the concentration we are trying to make. Let's start with the 2.5 because the 0 can kind of throw people off. V2, this is the volume of that final solution we want to make. It tells us right here we want to make 50 milliliters of solution. So what we have here is 20 times V1 equals 2.5 times 50. So V1 is going to equal 2.5 times 50 divided by 20.
$6.25.
Yes, that will be 6.25 milliliters. So that's what we would put here. And now for this next solution, all we're going to switch out, basically, we're going to have this, we're going to start with the same formula or this kind of same setup. And we're just going to switch out what we're making. So instead of making a 2.5 molar solution, we want to make 5 molar. And then it's just plug and chug. V1 equals 5 times 50 divided by 20.
So that would give us 12.5.
Well,
12.5, yeah, exactly. And I'm just gonna do the other ones here.
$18.75.
Mm-hmm. Yeah, 18.75 mils. Exactly.
I have that the last one is 25.
Yep, perfect. And then we didn't do this first one because sometimes if you put like a 0 in your formula, I don't know, sometimes students get confused with that when a 0 gets inserted in, but you can also just think about it logically that if you're making a 0 molar glycerol solution, so a solution that has absolutely no glycerol, then how much of your stock glycerol are you going to put into it? Nothing. So we don't want any glycerol in this first solution, so we're not going to put any amount of our stock into that dilution. And now we know the final volume of each of these solutions is going to be 50 mils. This is just 50 mils all down the line. And then we are going to use our final volume and then the volume of stock that we're adding to calculate how much water do we need to add to get this final volume. So if we have 0 amount of stock,
50.
yeah, it's just going to be pure water. And then for the next one,
50 minus 6.2.
this is, wait, 43.75. 27.5. We know this is 25. This one is This is 31.25, I think. Someone check my math. Yeah, that's right. All right, so very easy. Once you know what formula to use and how to use it, it just becomes a matter of, making sure you're entering it correctly into your calculator. So really straightforward mathematics there with these dilution calculations.
Professor, I have a quick question.
Yeah.
I need help with the 41. It's just that I'm putting the numbers in my calculator, but I'm not getting the answer right.
Oh, and 41?
Mm-hmm.
Okay. We'll start with, I'm not sure what kind of calculator you have, but start with this first operation. Just put 17 minus 15 and then hit enter.
Okay.
And
Okay.
then, so that should give you a 2. So then 2, and then divide that by 15, hit enter.
Okay.
And then
Okay.
multiply by 100.
Oh, okay.
You got it now?
Got it. Thank you.
Okay. Yeah. Yeah, because like sometimes, if you don't have the kind of calculator where you can put in these brackets and you just put it all in a row, the calculator then decides how, like in what order it's going to calculate things. And so that can definitely change your outcome. So just
thank you.
Take it a little bit at a time. So I'm not sure if everyone here was with us earlier, but in case anyone has not yet, signed into the digital sign-in sheet, I am copying that link now. There we go. So just make sure you sign in. If you've already signed in, don't worry about it. This is just for anyone who's dropped in since the last time that was shared. All right, so now we're moving on to, some more percent change. So this had to do with our potato pieces. So this is our raw data that's just showing the weight in grams. And we want to know what percent change or what the percent change in mass was, whether it gained or lost mass, and exactly how much of its total mass it gained or lost. So this basically just uses the same formula as up here. So we're talking about percent change. So the mass at some time minus initial mass divided by initial mass times 100. So it's going to use the exact same calculation or formula as up there. We don't need to go over all of those calculations unless you really want to. I think this question is more important of why we calculate percent change in mass. Why do we bother with this kind of transformation of the data rather than just using these raw values?
Because not all the potatoes are the same size.
Yeah, excellent, friend. Yeah, so first of all, not all the potatoes start out the same way. You might cut them in very similar, to very similar dimensions and try to make them all identical, but they're not always going to start out the same.
Also because not all the cells within the different potato plugs will be the same. So some of them might be like more hypotonic or hypertonic.
Okay, perfect. Yeah, that's another good point. So not all the cells within each potato piece are identical in their, tonicity or, I don't know, internal concentration of starch. Right, so there are like very minute differences between these different potato pieces that are placed in here, and we want to normalize it, or that's kind of the terminology that's used in research, is we say we want to normalize the data. We want to kind of clean it up and make it so that it's easier to compare these values, so that we can see at a glance, like what has gained weight, what has lost weight, and how much exactly in comparison to the other experimental pieces. So I guess I could sum that up by saying, it's easier to compare the results, the experimental results, across different potato pieces. When your data is normalized. All right, so for example, for this, well, like I said, 0 minutes, percent change is just 0, right? Because you're just 5.3 minus 5.3 divided by 5.3. So that would mean 0 times 100, which is 0. So when we normalize the data, we start out with a nice clean value. So every potato piece kind of has the same starting point numerically, and then it's easier to compare the rest of the results. So I'm just going to write out this one calculation really quick. So this would be 5 minus 5.3 divided by 5.3 times 100. So it is a little bit tedious to have to do these calculations for each, box in the table. But again, once you know your formula and how to use it, it's just a matter of making sure you're entering it into the calculator correctly to get the right answer. And that's actually another good reason why we ask you sometimes in these lab exam questions to write out the formula you're using. Because let's say you write out the formula correctly, but you just enter it into your calculator incorrectly and get the wrong answer. So if someone has a wrong answer, but they've set it up correctly, I still give them credit because they obviously know how to use the formula. And maybe it was just, oh, they moved a decimal place, or they, forgot to clear their calculator before inputting these numbers. There's a lot of things that can go wrong when you're putting in an answer. So if you just put the answer and the answer is wrong, I can't give credit for that. But if you put your formula set up and your calculation kind of written out this way, I can at least give credit for that, even if your actual answer is incorrect. That's how I grade. I can't speak for all of the 150 professors, but I don't know. It makes it easier to give partial credit if we can see what you're thinking and what your reasoning was in these questions. So let's move on to question 44. So if we were going to plot this data that we just calculated, what would the independent and dependent variables be?
The dependent variable would be the mass of the potato plug, and independent variable would be the time and the solution concentration.
Exactly, yeah. So we kind of have two independent variables. One would be time. So of course we're looking at this change over time. And then we also have these different solutions. So we'll say solution concentration. So our independent variables, one or both of them, are they quantitative? Can they be described with a number, time, and solution concentration?
Yes.
Yeah, absolutely. Are they continuous?
Yes.
Yes.
Yes, they
Well-
are. Yeah, they are both continuous. Time and concentration are continuous.
Why would the concentration be continuous if technically you kind of like you're assigning the concentration versus time is already something that occurs naturally?
Right. So by continuous, what that means is if there are an infinite possible number of data points in between the ones that you have chosen to measure. So time is continuous because, so between 0 minutes and 20 minutes, you know, those might be the times that we decide to measure the mass of this potato. But think about all the little increments that exist between 0 and 20 minutes. So you could look at it like minute by minute. So 0, 1, 2, 3, 4, 5, you know, all the way out to 20 minutes. Or we could choose to look at it every half a minute, you know, every 30 seconds or every 20 seconds or every half a second or every millisecond. You know, like you can keep kind of parsing down the time into smaller and smaller increments and to an infinite degree. So between 0 minutes and 20 minutes, even though we're just recording these 2 points in time, there are an infinite number of possible time points that could be measured between 0 and 20. And likewise between 20 and 40 and 40 and 60.
So what would be an example of a non-continuous independent variable?
So something that would be non-continuous.
Could that probably be like height?
Well, like even height could be, you could have an infinite number of measurements depending on if you're, you know, you can measure yourself in meters or in centimeters or millimeters or micrometers or nanometers.
Okay.
So, okay, Reem was saying different lakes. Yeah, so if you think back to that very first lab exercise that you did with all the graphing, and one of the graphs that you were asked to make was like the number of tadpoles per lake. So the lake, that was your independent variable. And obviously that's not a quantifiable variable, it's a category. And it's also not continuous. So there's not like an infinite number of lakes or things. Those are like discrete locations. Yeah, so I guess you could say location is not continuous.
So then quantitative variables are always continuous?
You know, I'm sure there exists some example of a quantitative variable that is not continuous, but by and large, I guess the examples that we've used in class, yeah, if it's quantitative, it's usually continuous.
Okay, thank you.
Because it's the same thing with solution concentration, is like here we're looking at molarity, but between these 2 molarities, you could look at the micromolarity or the nanomolarity or the picomolarity, and there are infinite number of molarities that could be measured in between these 2 points. So yeah, that's what makes it continuous.
Okay, thank you.
So if it is quantitative and continuous, what kind of graph would we make?
A line graph.
Yeah, exactly. Line graph is the way to go for continuous and quantitative data. Yeah, so if your independent variables are both quantitative, I always misspell that word, quantitative and continuous. And it asks you to make a graph, although realistically on your lab exam, because it's being given through Blackboard, you're not going to have to make an exam or a graph on the exam. You will be asked to interpret graphs. So you'll be shown a graph and be asked, you know, about the results, kind of like the sunscreen example we saw earlier. but you would not be expected to actually like produce a graph because we just don't have the capability to do that in an exam setting, at least not digitally. All right, so now it is asking us for the rate of percent change, based on our graph, which we didn't make. So if we had a graph of these different concentrations and our change in mass of your potato, so what would we expect that graph to look like? We know it'd be a line graph. And we know that on our y-axis, we would have the mass change, percent change in mass. On the x-axis, we would have our time, and then we'd have different lines kind of signifying these different solutions. So this is similar to what you did in your lab reports that you just turned in. So you'd have different lines, maybe one going that way, maybe another one going like that, another one going like that. Wait, how many solutions do we have? I think there were, oops.
Yeah, let's not get it.
So yeah, let's pretend that this is like our graph, and then we would want to calculate the slope of each line. So if you're doing that manually, if you've just produced a graph with these lines, hopefully everyone remembers this from some kind of geometry class. So the rate of percent change, how fast things are changing, either increasing or decreasing in mass, is you take 2 points on the line, and then you have your y value minus your other y value, x value minus x value.
So you can take either 2 points?
So this, by 2 points, I mean like you would, so for this line, you would probably, the easiest way or the most, I guess, accurate way to calculate the slope would be to use your starting point and then your end point.
Okay.
So the starting point for all of them, I think, is 0, 0, right? 0 minutes, 0 change in mass. And then you would do the 60-minute change in mass. So your coordinates, right? Because coordinates are always written as x comma y when you're talking about points in a graph. So your first point is going to be 0, 0. That's the origin of your line. So at time 0, you have 0% change. That's basically what that means. And then at your second point, this would be at 60 minutes. What would the change in mass be? I guess we could go back up here and calculate that. So this would be 5.8 minus 5.3 divided by 5.3 times 100. And I got 9.43%. So that would be the other part of this value. Say 9.43 or 34. 43. Okay. So remember, X is our time, Y is percent change. So at 0 time, we have 0% change. At 60 minutes, we have 9.43% change. And then we would set it up this way. So Y2, that's 9.43 minus Y1. Well, Y1 is 0. And then divide that by 60 minus 0. So that makes it really easy, actually. So our slope is 9.43 divided by 60.
0.16.
0.16. All right, nice. So that is our rate of change. All right, how are we feeling with that one? Do we want to do the next one as well?
I guess maybe we could do one more.
Okay. All right, let's do the 0.2 molar glucose. So we don't need to calculate all of these boxes. We already have our 0 time, 0% change. So all we need to do is the 60-minute one. So let's calculate that and then calculate the slope. And hopefully we come up with the same answer. Thank you for speaking for the group, Diana.
I got 1.75.
I did too. Excellent. So 1.75% change. All right, so now if we calculate our slope.
I got 0.029.
Excellent. Yeah, so did I. So that's perfect. And remember that, you know, if it's a positive percent change in mass, that's indicating that it gained mass. It could also be a negative value too. If it was a negative change in mass, that would indicate that your potato piece lost weight. So the slope could also be negative. So if one of these values happens to be negative, then you'd end up with a negative slope, and that's perfectly fine. All right, so if we wanted to plot the rate of change, which we just calculated up here, so the rate of change versus glucose, how would we do that? So what would be on the y-axis as our dependent variable?
So the rate of change depends on the glucose concentration.
Yes.
So the rate of change would be the dependent and the glucose concentration would be the independent variable.
Exactly. Yeah. Glucose concentration. Yeah. So how quickly that potato loses or gains water is going to depend on basically, yeah, how concentrated that solution is, how hypertonic or hypotonic it is. So glucose concentration, is it quantitative? I'd say so. Is it continuous?
Yes.
Yes.
Yep, it is. Right, so because of that, we would be making a line graph. And then, of course, it asks you to make a graph, and this is going to be that graph that kind of like the graph number 2 that you had to make for your lab report. So it started up here, it crossed the x-axis, and went down here. And there was something really significant about where it crossed the x-axis.
So, Professor, the y-axis is the dependent variable or the independent variable?
Y is the dependent.
Okay, and then the x-axis is always independent.
Correct, yep. Yeah, so we had glucose concentration here and then rate of change here. So at first the rate of change was positive because those pieces were gaining weight. And at a certain concentration, the potato pieces started losing weight. So what was special about this point right here?
It's the isotonic point.
Yeah, exactly. Yeah, so where this particular line crossed the x-axis, this was our isotonic point, where the concentration of solute inside the potato cells equals the concentration outside. So that's what was special there. And I think that's what it's getting at for this question 46, that what can you tell from this graph that you couldn't from the previous graph? That would be your isotonic point.
Professor, and you said that the isotonic point is where, I guess, like the line crosses the x-axis?
Mm-hmm.
Okay.
So the isotonic point, that's our concentration at which the potato piece does not gain or lose weight, because those potato cells are now isotonic. They're in equilibrium with the solution around them.
Professor, so for example, when you have the potato plug like gaining mass and then you have the potato plug losing mass, will both, I guess, like, will in both instances, will they continue to gain and will they continue to lose until they reach the isotonic point? So like, is that the point of the experiment or?
So yeah, theoretically, if you left those potato pieces in the solution long enough, I mean, as long as there's enough fluid within the cells to be drawn out and like dilute the solution outside of it, theoretically, it is trying to achieve that isotonic situation. So it would keep losing water or gaining water to reach an isotonic point.
Okay, all right. Because I was confused. I thought that some potatoes were hypertonic, others were hypotonic, and then others were isotonic. But no, the goal of submerging them is to reach their isotonic point.
Right.
Okay.
Yeah, and then like the purpose of like this whole experimental setup and all these calculations was, rather than like breaking open a single potato cell and trying to figure out what the concentration of solute is inside of those cells, we can kind of indirectly determine what the isotonic point is by having a bunch of different pieces in different solutions, doing some math, and then that's how we can calculate that concentration of solute inside the cell. Oh geez, another hour just flew by. Okay, we are getting ready for some cell respiration. And I think this is the last little bit. Yeah, so it's just these 2-3 pages. So I'm going to say let's just power on through and finish this up. And then we can take another break after cell respiration. And then I'll just kind of open the floor up to like any questions in general, or like if you want to go revisit something, we can do that as well. All right. So cell respiration, this was oh, photosynthesis. Okay, how about we stop? I don't know. We'll do photosynthesis. We'll do cell respiration and then take a break and then do photosynthesis. Just because I don't want to overwhelm anyone's brains. Okay, so aerobic respiration or cellular respiration, very important cellular process. Where does it take place? Don't worry about how many ATPs are produced. No one knows for sure. I don't know why they put that question on here. Where does cellular respiration happen? Why is it important?
Mitochondria.
Cytosol.
Yeah, it happens in the mitochondria. It's about transforming energy. What kind of energy?
ATP.
What's that?
ATP.
Okay, so we know we want to get ATP. So we're a process of transforming energy from
Glucose.
something to ATP.
Glucose.
Yeah, glucose or really any organic fuel. Of course, we always use glucose because it's a nice, easy starting molecule to explore these topics, but any organic molecules really. So we're transforming energy from glucose or any organic molecule. This is one type of chemical energy, and we're transforming it into another type of chemical energy, one that is readily usable by our cells and their processes. And how does this differ from fermentation? What is fermentation?
It's an anaerobic process.
Right, anaerobic process, which means no O2. So that could be an advantage of it, right? Advantage is you don't require oxygen. Do you need organelles to do fermentation?
No.
No, you don't. This can all happen right in the cytosol. So this is why bacteria and yeast and those kinds of organisms can do fermentation. So you don't need any kind of compartmentalization to do this. So anyone can do it. Anyone can do fermentation. That's one of the advantages. Don't need oxygen, don't need organelles, don't need any fancy equipment. What would a disadvantage be?
Low amount of ATP.
Right, it's a very low yield of ATP compared to aerobic respiration. So that's kind of the major drawback of it. And I feel like that pretty much sums up what the differences are between cell respiration and fermentation. So one needs oxygen and organelles and you get a whole lot of ATP. The other one doesn't need oxygen or organelles, you get far less ATP. Okay, our summary equation. So we know we need glucose. What's our chemical formula for glucose?
CH2O.
C6.
Yeah.
H12O6.
O6, excellent. Karen, I heard you mention H2O, or I don't know if I heard you get cut off or not. So H2O is going to be one of our products. So we're going to put that over here. What else do we need for cell respiration? We kind of already established it up here.
O2?
Yeah, we need oxygen. Absolutely. You stop breathing and your cells stop being able to do respiration pretty quickly. All right, what are the other outputs? So we know water is an output.
Carbon dioxide.
Yeah, carbon dioxide. And of course, we're going to get ATP plus a little bit of heat. So some of that energy gets lost as heat. How did we determine respiration in that experiment with this device?
Because of the amount of O2 that was consumed over time.
Right, volume of O2 consumed. Right, so this little guy was using up oxygen, and as he used up oxygen, what happened?
The water was moving inside the pipette.
Yeah, exactly. Yeah, so that's why we had to put this underwater. So when we first placed it underwater, we had some gas pressure here and in this pipette, which kind of held the water out. But as he's using oxygen, as he's doing cell respiration, that water starts to creep in. And as that water creeps in, that's what we actually measure using the little lines marked on the pipette. So the respirometer was submerged to measure, the volume of oxygen consumed on the pipette. What was the purpose of KOH?
I believe it's to take away carbon dioxide.
Right, so KOH, so it eliminated the carbon dioxide from the experiment or from the setup. And how did it do that? How does KOH eliminate the CO2? Right, by turning it into a precipitate, a solid, with the formula K2CO3. Was it potassium carbonate, I think is what it was called? Right, so we didn't want to have any CO2 produced because that would kind of keep this gas pressure stable. We wanted to eliminate that so that we can actually see the oxygen being used up. Yes, Jenna, this review is being recorded. So once I'm done here, I'm going to upload it to YouTube, which will probably take an hour or two, but I don't think anyone's going to be like rewatching this exactly tonight. I mean, you have like a whole week and a half before your lab exams. So, but yeah, this will be posted and I'll share the link with all your professors so everyone will have access to this recording. All right, so we didn't just use the crickets in this experiment, we also used peas, some of which were germinating. What is germination? Or basically another way to ask that is, how are these seeds staying alive without leaves and chloroplast, and how can they be alive and not do photosynthesis?
It has starch inside them.
Right, yeah, so peas and all seeds contain starch, which is our stored form of glucose. And they use it for what?
Growth.
What chemical process are they doing?
Photosynthesis.
Nope, not with the starch. What are they using the starch for?
Energy.
Yeah, and how are they getting that energy?
cellular respiration?
Yes. Yeah, these peas that are germinating, they're doing cell respiration, just like that cricket. So the seeds have to rely on cell respiration because they don't have leaves yet. They don't have any chloroplasts. They haven't produced those organs to capture light energy. So they have to rely on cell respiration if, you know, they actually want to grow into a plant. So that's how seeds stay alive.
Just out of curiosity, when you plant a seed and it doesn't grow, does that mean that it wasn't able to carry out cellular respiration, or is that like human error?
It could be a lot of different things. So yeah, it could be that for whatever reason, it couldn't carry out cell respiration, like maybe some of the organelles were damaged. Because again, in order to do cell respiration, you need mitochondria. So, you know, maybe some of the internal workings of the cell were damaged, or yeah, maybe it didn't get enough oxygen, or maybe it got too much water, or it got too cold. Could be any number of things. So not necessarily human error.
Okay, thank you.
All right, so I see that we have some other folks, I think, who have joined. So I'm going to share the link again in the chat for recording your attendance. So just write your name, your M number, your instructor's name, whoever teaches you 150, and then which review you're in. All right, ooh, calculating metabolic rate. So this is, again, one of the formulas that you'll need to know for the exam. And similar to how this question is set up, it just gives you some values, some experimental data, and it asks you to calculate a metabolic rate. So let's start with what are the units for metabolic rate? In what units are we measuring metabolic rate? So we know it has something to do with consuming oxygen, right? Because that's one of the things we measured. So milliliters of oxygen per hour. So there's another parameter that is taken into account when calculating metabolic rate. So
Grams?
the milliliters per, yes, grams. Milliliters per hour is just the respiration rate, but the metabolic rate takes into account the mass of an organism or an object or whatever. So it's milliliters of O2 per hour per gram. Okay, so now that we know what our units are, this will help us kind of extrapolate what we need from this. Oh, why am I down here? Whoops.
So Professor, if you were to have minutes instead of hours and then kilograms instead of grams, you would have to convert from minutes to hours and kilograms to grams, right? To be able to use the formula?
Correct, yeah.
Okay.
Yeah, so we have, so it's talking about 2 mils of oxygen in 6 minutes. So let's convert this to milliliters per hour. All right, so if we have, so 2 mils per 6 minutes. Okay, and now we want to eliminate that minutes unit and get it into an hour unit. So we're going to multiply that by 60 minutes per hour. Okay, I know it's not set up right here because it's linear and it's not, doesn't look like a nice kind of cross multiply thing, but 2 mils per 6 minute. Times 60 minutes per hour. So then the minutes cancel out and we're going to be left with mils per hour. So let's do this calculation. and see what it comes to.
Would it be 0.1 hours?
Times 60. You said 0.1? That is not what I got. Hold on.
12 minutes.
Well, I'm getting 2 minutes times. I'm getting 20 mils per hour.
I got the same.
Okay. So 2 divided by 6, that was like 0.33333. And if you multiply that by 60, you should get 20. So now our minutes units have canceled out. Let's see, can I do a little strike through? Nope. Okay, never mind. So now 20 mils per hour. So that's our respiration rate. But we want metabolic rate, which takes into account the size of an organism. Oops. Okay, so we have mils per hour. Now we have 10 crickets that weighed 2.5 grams. So if 10 crickets weighed 2.5 grams, do they want it for 1 cricket? Probably. So if 10 crickets weigh 2.5 grams, how much does 1 cricket weigh?
0.25.
Right, 0.25, basically. Right, so if we have our metabolic rate, oops. So 20 mils per hour divided by 0.25 grams. And what does that come to? So basically 20 divided by 0.25.
If you did that, would you have to divide the 2 milliliters of oxygen by 10 as well to get the individual consumed rate per cricket?
Okay, so I guess there's 2 ways we could do that. So yeah, I also got 80. Okay, so we either divide by 10 in the grams or after we've calculated metabolic rate, if that makes sense. So let's try it this way where we have 2.5 grams, and then whatever this answer is, divide that by 10 crickets and see what the answer is. Oh yeah, that changes the answer a lot.
Professor, Dr. Smith's answer was 8 milliliters per hour per gram.
2 mils, 60 minutes divided by 2.5 grams. Okay, so we don't need to take into account the individual crickets. Okay.
Right. He just did 20 divided by 2.5.
All right, so yeah, then it would be 8 mils per hour per gram. So respiration rate, again, is milliliters of oxygen per hour. When you take into account the weight or the mass of what's consuming that oxygen, then it becomes metabolic rate. All right. Okay, so it's 6:45 now. We have 1, 2, okay, so basically like 2 whole pages left. I don't know about you, but I need a little break. So let's do, so it's 6:46. Let's come back at 6:57. That's 11 minutes. 6:57, and then we will finish up this review. And then I'll take questions. All right, I'm going to pause the recording and I'll see you at 6:57. Resume the recording. Great. All right, let's finish up this document. So we have a little bit more about metabolism. So it's asking us about the relationship between body size and metabolic rate. So would you expect a larger animal to have a higher metabolic rate or a smaller animal to have higher metabolic rate?
When the size increases, the metabolic rate decreases.
Yeah, that's generally the trend that we see. So as body size increases, metabolic rate decreases. And it's thought to have something to do with the kind of surface area to volume ratio of organisms. But if we are comparing 2 different types of animals, so for example, a reptile, which is an ectotherm, and a mammal, that's an endotherm, and if we controlled for their mass, so if they had the exact same mass and they're both being held at the same temperature, what would you expect to see? Who would have a higher metabolic rate and why?
It will be the mammal.
Right, so mammal would be expected to have higher metabolic rate. Why is that?
They're an endotherm.
Right, because they are endotherms, and what do they use that extra metabolism for?
To produce heat.
Yeah. Yeah, so they need to maintain a stable body temperature. So they need to be doing cellular respiration a lot more quickly and a lot more consistently in order to maintain that body heat. So remember that every time we make ATP with cell respiration, we are producing a little bit of heat. Some of that energy doesn't get transferred over to the ATP molecules and it's just kind of dissipated out into the environment. Although endotherms have special body modifications to help trap that heat, and that's how we maintain our internal temperature. So now it's asked us to look at some data from an experiment where we were looking at the respiration, so milliliters of oxygen per gram per minute. Okay, this seems like a riff on the metabolic rate that we calculated earlier. But anyway, we're looking at a cricket and a mouse, so just how much oxygen they're consuming per unit of their mass and at 2 different temperatures. So 10 degrees Celsius, that's pretty chilly. That's about 40 degrees Fahrenheit. 25 degrees Celsius, that's closer to room temperature. That's like, I don't know, 74 degrees Fahrenheit. So according to this data, the crickets at 25 degrees have a greater oxygen consumption per gram than at 10 degrees. And then this is opposite of the trend that we see in mice. So why do we see this difference in their rates of consuming oxygen at different temperatures?
The mice is an endotherm, so the warmer it is, the less energy they have to use for heat.
Yeah, exactly. So yeah, the mice are the endotherm. So when we increase the temperature, they don't need to expend as much energy keeping their bodies warm. What about for the crickets? Why does this increase? So crickets, they're not endotherms, right? They're ectotherms, and their metabolism is basically at the mercy of their external environments. So if the external environment is cold, they're not doing a whole heck of a lot of cell respiration. If the external environment is hot, then they're going to do more respiration. So when the temperature increases, so does their metabolism. But for an endotherm that already has its own heat source, it has an opposite trend. So the difference between these two is their mode of temperature regulation. And that's basically the answer to this question 56. So the relationship between metabolism and surrounding temperature. So in ectotherms, metabolic rate will increase or decrease along with temperature. You could say it's a positive linear relationship, or it's a linear relationship. And in homeotherms, also called endotherms, the trend is reverse.
Wait, in ectotherms, is it positive or negative linear, or is it positive?
Wouldn't it be a negative linear trend?
Because as temperature increases, their consumption of oxygen increases.
No, for ectotherms would be the mouse, right?
No, the mouse is the endotherm.
Okay. I was confused. So then the endotherms would have a negative linear relationship.
Yeah, it's a little bit more complicated. It's not quite as cut and dry as they're presenting it. Like for the most part, endotherms have a pretty stable metabolic rate, and the changes that you see according to temperature are much smaller compared to the changes you see in the ectotherm. Like the ectotherm's metabolic rate is very tightly linked to the external temperature. So for endotherms like us, our metabolic rate is pretty stable. In warmer temperatures, I meant to say, might decrease a little if it's really hot outside. It might slightly increase if we're out in like cold for a long time. So there is like a negative trend, like a negative linear relationship, but with less deviations, I guess you could say. So yeah, we'll say it's a negative linear relationship, but there's less deviations.
But I'm still confused about the ectotherm. So does it go both ways? Is it positive and negative? Like, because if the temperature is decreasing, doesn't the respiration also decrease with it?
So I guess it depends on how you're looking at your graph, because remember we like to read graphs from left to right. So if we had a graph here, and this is temperature, so temperature is increasing from here to here, and then this is oxygen consumption of the cricket. So as temperature increases, his consumption of oxygen is going to increase as well. So that would be positive linear. If for some reason you reversed your x-axis for some reason, I guess then it would be a negative relationship. Or like if you started at a hot temperature and then you decrease the temperature steadily, the metabolism or a respiration rate would also increase or also decrease along with temperature.
Right. So the second one is kind of what I was thinking. So like if it was high and went to low, that's negative, right?
Yeah. So maybe just to clear up, we'll just say it's a linear relationship. So as one variable increases, so does the other. If this variable decreases, so does this one.
Yeah, that makes more sense to me. Okay, thank you.
Okay. Okay. Yeah, so there are some questions on the lab exam about the difference between ectotherms and endotherms. Trying to think if there's something kind of like this. Probably, yeah, at least one of these questions where you have to kind of interpret the data that's given to you and explain why that there's a difference. So just make sure you understand that core difference between ectotherms and endotherms. And actually, if you go to the, what was that? That was exercise 5, the lab handout. If you go to that lab handout, there's a link to a YouTube video in the lab handout that does a really good job at explaining the difference between ectotherms and endotherms. So I would say go back and watch that maybe, and hopefully that'll like kind of clear up any misconceptions you might have. And yeah, just as another way to kind of take in that information. So that's in the lab handout. It's not here in the review packet, but I would recommend watching that. I think it's like 10 or 12 minutes long. All right, home stretch. We are talking about photosynthesis. So this is the lab exercise we all just did last week, so it should be fresh in our minds. So what is the point of photosynthesis? We're going to be transforming energy, but what kind of energy do we start with and what kind of energy are we ending up with?
Light energy?
Yeah, exactly. So it's the process of transforming light energy from the sun into
Chemical energy?
Yeah, chemical energy, exactly. Like glucose and other sugars, other organic molecules. And it's carried out in those chloroplasts. Oh, sorry, which organ of the plant? So we know that organelle is the chloroplast. What do we mean by organ?
Leaves?
Yeah, leaves. So by organ, we mean the leaves of the plant. That's what contains the majority of the cells that make chloroplast and chlorophyll and have all the important parts for carrying out that process. Photosynthesis, and of course we know cell respiration is happening in the mitochondria. All right, our summary equation. Ooh, it even gives you a little word bank here. All right, so what are the inputs for photosynthesis? So yeah, we know we're going to need some carbon dioxide.
Water.
Yes, absolutely. Water. H2O.
Light.
Yeah, we need our light energy. And then what are the two outputs? Ooh, thank you, Reem. Okay, so then we end up with our glucose, C6H12O6, and of course, oxygen is a byproduct. All right, in the second of the 3 activities of that lab exercise, there was a separation of those pigments in chloroplasts. How many pigments were ultimately shown there?
3.
3. So, oh, I think there were 4, but 2 of them are kind of related. So can we name these pigments?
Carotenes.
Yeah, carotenes, those are like the yellowy ones.
Chlorophyll A and B?
Right, chlorophyll A and B, which they're related. They're very similar in their structure, but they're still technically 2 different molecules. So they count as separate ones. And then there's a last one. It's okay if you don't know how to pronounce it. They're called xanthophylls. Xanthophylls. And where are they located? So not just, okay, in the chloroplast, but specifically, where are these molecules kind of embedded? Oh, thank you, Bezoit. Yeah, so they are embedded in the thylakoid membrane. Right, so within the chloroplast, you have like these stacks. They almost look like pancakes or coins or something. These little discs all stacked up. And they're basically just stacks of like these membranes. And so within that membrane is where you'll find the pigment molecules. So the pigments in most plants absorb what colors? What were the colors that were
Blue.
most readily absorbed?
Blue and red.
Yeah, so kind of a violet blue color. Ooh, and that's small. And red. Yeah, so those 2 ends of the spectrum. Oh my goodness, what is this statement? Okay, plants perform highest photosynthesis by using wavelengths of, so between the violet blue and the red, so which one did it prefer?
Violet.
Yeah. Violet blue. Does anyone remember the wavelength, the number of the wavelength of this peak?
Was that like the 300s, 400s? No.
Yeah, this was like the 400s. So it's like 400, 420, somewhere around there, that color of light. And here, because it is absorbed best. So that's why you get the most photosynthesis, because it's mostly absorbed, very little of it, if any at all, gets reflected or transmitted. Wavelengths of this color contain highest energy. Yes, thank you, Reem. Right, so at the violet spectrum, the wavelength is smaller, right? We have values of like 400 to 420. At the red end of the spectrum, we're talking about wavelengths of like 680, 700. So because the waves are closer together in the violet blue spectrum, that means those the light has more energy because the waveforms are closer together instead of being more spread out. So let's actually, I want to write that. So that's an important relationship to keep in mind. The shorter the wavelength, the higher the energy. Oh, geez. Okay, so what does photosynthesis do for us? Why is it so important?
Provides oxygen.
Right, so it produces oxygen for us aerobic organisms, as well as what's its other product?
Macromolecules.
Yeah, organic molecules like our glucose. So it produces those things, oops, which we require organisms require, yeah, so it helps, it removes carbon dioxide from the atmosphere, which is great. Why is chlorophyll green?
Gets reflected or not absorbed.
Right, because yeah, that particular wavelength is not absorbed, it is reflected. So it comes back to our eyeballs, and that's why we sense leaves as being green, at least in the springtime. The absorption spectra. So this was the graph that you made in the third part of the third part of the lab that I don't want to underline it. Okay, there we go. The absorption spectra, this is the graph that you made, which was like absorbance versus wavelength. So what did that tell us about chlorophyll pigment? So this is the graph that was absorbance versus wavelength of light. So we passed different wavelengths of light through this sample of chloroplast, and then this machine, the spectrophotometer, measured how much light was being absorbed, and that's like the number it spat out at us. So that absorption spectrum indicated which wavelengths were absorbed the most or the least. Okay, we got a couple more questions here. So this one is about the paper chromatography. So this was the separation of pigments. So we found that the solvent traveled 20 centimeters from the origin. A pigment molecule traveled 17 centimeters. What is the RF value? How do we find that out?
Divide 17 by 20.
Yeah, exactly. So it's the distance traveled by the solvent, or sorry, the pigment, which in this case is 17, divided by distance traveled by the solvent. So what does that come out to, Ephraim? Oh, I see Reem has already told me, and so has Kadiatu. Thank you. 0.85. That is our RF value. Very nice. Okay, so this is again one of the equations you'll need to know for the lab exam, so make sure you have that memorized. You know how to use it. This one's pretty straightforward. All right, the first of those three experiments in the photosynthesis lab exercise, you're measuring the rate of photosynthesis. How do we calculate the rate? What output or input did we measure? Ephraim, you're on mute. It looks like you're talking, but you're on mute.
Oxygen consumption?
Not consumption.
Or production?
Yeah, oxygen production, exactly. So how quickly it could produce 0.1 mils of oxygen. And this was the setup where we had like the tube, a plant in the tube with like a bright light shining on it. And then we were measuring these little oxygen bubbles that were coming up into a pipette. So we used a solution of sodium bicarb in that experimental setup. Why? Why was the water in which that plant was sitting in, why did it have this baking soda or sodium bicarb in it?
To produce CO2.
Right, it was our source of carbon, which is a necessary component of photosynthesis because it's part of that summary equation, right? So without that, we could have shown as much light as we wanted to on that plant. It wouldn't have done any photosynthesis without a source of carbon in there. All right, so we have some data produced from a similar experiment where we have different wattages of light bulb and the time it took to produce 0.1 mils of oxygen. So this is in minutes, but we want to calculate the oxygen produced per hour. So how would we set that up? Who can kind of walk me through how you would set up that calculation?
So you would start by converting the minutes into hours.
Mm-hmm.
So you would divide 30 by 60, which would be 0.5 hours.
Right, so 0.5 hours, yep.
And then I guess you would do the 0.1 milliliters over 0.5.
Yeah, beautiful.
And that would give you 0.2.
All right, so 0.2 mils per hour. That's our first rate. All right, let's do, if it's 15, it's going to be what, 0.25? Nope. 0.4 and so on. Okay, so that's pretty straightforward as well. So once you have your units all figured out, then it's really easy to calculate this rate of photosynthesis. Oh, and then it wants you to make a graph of it. If we were to make a graph of this, what would be on the x-axis? What would be on the y-axis?
Light intensity would be on the x-axis and the rate of photosynthesis would be on the y-axis.
Yeah. So the rate of photosynthesis is dependent on, yeah, the wattage of the bulb. So that sounds good to me. Yeah. So if we were to graph this wattage on the x, rate on the y, and we would be able to kind of visualize how the increase in power of the light changes this rate of photosynthesis. All right, so I guess that is it for this, at least going over the answers to this document. So we still have technically a half hour left. I'm obligated to stay here for that time. Of course, you're free to leave if you've had all your questions answered. Thank you for attending. I'm going to post one more time the link for recording attendance. So in case anyone has not yet clicked on the link and filled out their information, please make sure you do so. You're welcome, Karen. I'm glad that you found this helpful. So yeah, I'm going to stick around and I'll answer any questions you have. I can tell you about how many questions are on the exam, the formulas. We can go over the formulas again that you'll need to know, those kind of little details.
How many questions are on the exam?
34. So we have 34 questions and it's worth 120 points roughly. Although, yeah, like we get kind of like the base exam and we are allowed to modify it within reason. So if your professor wanted to add in a question or take out a question, they have that liberty. But round about 34 questions and 120 points. And you are very welcome, people thanking me in the chat. My pleasure.
Yes, thank you. This was very, very helpful.
Good, good. I'm glad.
Yeah, thank you, Professor.
Professor, where can you find the recorded review session?
So I'm going to be uploading this to YouTube. That's going to take a little while. Once it's uploaded and kind of ready for viewing, then I'll send out a link. I'll probably put it in an announcement or an email to the class. And I'll make sure that the other Biology 150 instructors get it as well. So if you're not one of my students, you should still be getting the link from your professors.
So I have a question. Well, it's not really like for me, but my sister, she's also in bio right now, and she's doing an osmosis lab. But one of her questions, like it particularly got me, I wasn't able to help her. But it's
so for our lab, we had to do a osmosis of red blood cells. Okay. And through the osmosis, one of the solutions that we had to make was with the blood and deionized water. And in the experiment, the deionized water was considered a hypotonic solution. So one of the questions is explain why water is considered to be a hypotonic solution. And I just don't really understand why DI water is supposed to be hypotonic.
Yeah, so deionized water, that's like an especially like clean kind of filtered water. And so it means like there's literally nothing in it but H2O molecules. So there's no random like sodium or chlorine or calcium ions. It's like as pure water as you can get. So it's hypotonic because it's just plain water. There's nothing dissolved in it. So that's why, especially if you put an animal cell into just pure water, that cell is going to, what's it going to do? Yeah, that cell's going to burst pretty quickly. It's going to lyse almost instantly because there's so much stuff inside of a cell. You know, there's all kinds of ions and molecules. It has a pretty high molarity. And plain water, it's just 0 molar. There's nothing there.
Okay. Thank you.
Mm-hmm.
Thank you, Professor.
You're welcome. So Efrain, I see your question in the chat if the guide will be similar to the actual exam. None of the questions are going to be like verbatim, you know, like copy pasted, but I guess the theme behind several of these questions you will see in the exam, or, you know, certain ones where they ask you about, they give you some data and ask you to calculate the numbers that you see on the exam might be different, but the equation you have to use to get your answer is going to be the same. So these numbers might be different. The little story it tells you about the experiment might be a little different, but you'll have to use kind of the same skills to get your answer.
Professor, are we going to have access to this document that you filled out?
Oh, I mean, yeah, I'll make this available. I have no problem with that. I'll save it and maybe I'll just like send it out along with my link to the recording. Because actually there is an answer key for this review, and I think most of the professors are posting the answer key tonight because this is the last review session. So You should have access to like the official answer key, but if you want like this specific document, I could send that out as well.
Okay, thank you. It's just because I did fill it out along with you. But there are like certain texts and drawings that you added that my computer doesn't have the capability to add, so I didn't add those.
Right. Okay. Yeah, I'll send this along.
Thank you.
Mhm.
Have a good night, Professor. Thank you so much.
You're welcome. You too.
Thank you, Professor.
You're welcome, Samita.
Thank you, Professor.
You're welcome. Oh, good. I'm glad that is helpful.
Professor, it's Chanel. I just wanted to see if we're going to have access to see what we got wrong on the test.
Yes. Yeah. So I haven't finished grading them all yet. But I do have like a setting of when the answers are going to be available to you or when you'll go back and review them. And I think I was going to do that like at the beginning of like the week when you come back from spring break.
Okay.
So if you can stand to wait that long, because I figured, yeah, like I'll send out an announcement or something, or I will announce it in class so that everyone will know like that information's up and available.
Okay. Thank you. I really appreciate you doing this tonight. I'm glad that I came to yours because you gave very like a different perspective on the answers than the other teachers did.
Okay.
Like one of the teachers, when they were doing like the hypothesis, it literally was like, this sunscreen is better than the other one. And I was like, Dr. Etz wouldn't say that. Like, I was like, she would not approve of that hypothesis.
Oh, that's funny. Yeah, it's yeah, I guess everyone has their own way of working through things, but I'm glad that my style is helpful.
Okay, thank you. Have a good spring break.
Thanks, you too.
Okay, bye.